morning, my dear students. Welcome to our Zoology online class. Dear students, hope you all studied the last class concept. And in the last class, we studied about two important concepts about this transcription. Okay, the experimental. We studied about two important concepts about the replication. Okay, the experimental proof for DNA replication. Okay, so this is one of the very important concept, and the second concept is the mechanism of replication. So these two topics we have completed in the last class. So hope you all studied these two topics. Okay, dear students, today we are going to study about one of the very important 5-mark question of this chapter. Okay, so listen very carefully, dear students. So today we are going to study about one of the very important topic. The topic name is called transcription. The topic name is called transcription. Here, the word transcription meaning the copying of genetic information from one strand of the DNA molecule into RNA is called transcription. The process of transferring the genetic information from one strand of the DNA molecule into a RNA is called transcription. Understand of you? This transcription process takes place with the process of one very important enzyme. Name of the enzyme is called DNA dependent RNA polymerase. So DNA dependent RNA polymerase enzyme playing a very important role in the transcription process. Okay, dear students. So now I said the meaning or the definition for the word transcription. Transcription meaning the process of copying the genetic information from one strand of the DNA into RNA is called transcription. Okay, so first we are going to study about the structure of a transcription unit. A transcription unit means a small area of the DNA molecule where the transcription process takes place. Okay, so that area is called a transcription unit. Actually, the transcription unit consists of three important regions. One is the promoter region, second one is the terminator region, and the third one is the structural region. So these are the three important regions of the transcription unit. Listen very carefully. Okay, here the first one is promoter region. This promoter region is present in the 5' end of a DNA molecule. Okay, in the two ends are there. One end is called 3' end and the second end is called 5' end. Here, this promoter is located or present in the 5' end of a DNA molecule. Okay, and this promoter region only provide the binding site for RNA polymerase. Okay, now we said one enzyme name DNA dependent RNA polymerase. So this DNA dependent RNA polymerase enzyme binding in the promoter region for the transcription process. Then next point, terminator. This terminator region is present in the 3' end of a DNA molecule. Okay, so in the 5' end promoter region is there, and in the 3' end terminator region is there. Okay, so in this region only the RNA polymerase stop the transcription process. Stop or terminate the transcription process. So hope you are understood these 2 points. Then in next point, in eukaryotic organism, the promoter region consists of more amount of adenine and thymine. So this region is called TATA box. This region is called TATA box. So in eukaryotic organism, the promoter region consists of more amount of AT rich region. More amount of AT is present. So this region is called TATA box. The same area, same AT rich region present in a prokaryotic organism is called Pripnov box. Is called Pripnov box. So the AT rich region present in the promoter region of the eukaryotic organism is called TATA box. The AT region present in the promoter region of the eukaryotic organism is called Pripnov box. So hope you are understood. So this is one of the 2-mark questions. So what is in the TATA box and what is in the Pripnov box? Okay. Then next point. So now we completed 2 important regions. But already I told you 3 regions are there: promoter region, terminator region, structural region. So now we completed the promoter region and terminator region. So now we are going to study about the structural gene. Actually, this structural gene have opposite polarity. Okay, 2 polarities are there. Then here always the transcription process in the DNA molecule, the transcription process takes place in only one direction. Okay, only one direction, maximum 3 to 5 direction. Okay, 3 and from the 3 and to 5 and only the transcription process takes place. So this strand 3 dash 5 dash strand is called template strand, and opposite strand 5 dash 3 dash end is called coding strand. Okay, so the structural gene having some opposite polarities. So always the transcription process takes place in from the 3 to 5 dash end. So this 3 to 5 dash end strand is called template strand, and opposite strand is called coding strand. Opposite strand meaning here 5 to 3 dash end. Okay, so hope you all understood this point. Then the structural gene, there are two types of structural gene is there. Okay, the structural gene may be monocystonic structural gene or polycystonic structural gene. Monocystonic structural gene or also two types of structural genes are there. In eukaryotic organism, the mRNA molecule carrying only a single gene, carry only one gene. So that single gene is code or responsible for the production of protein synthesis. Okay, so that mRNA is called monocystonic mRNA. So once I repeat this point, in eukaryotic organism, mRNA molecule carrying single gene, only one gene. So that single gene only responsible for the production of protein. So that mRNA is called monocystonic mRNA. In eukaryotic organism, a cluster of genes means it is always called operon. Okay, a cluster, a group of related genes are togetherly involved in the transcription process. So that mRNA is called polycystonic mRNA. That mRNA is called polycystonic mRNA. So I think you understood the difference between the monocystonic and polycystonic mRNAs. Understand, my dear students? Understand? Then next point about this transcription unit is the RNA polymerase. DNA dependent RNA polymerase consists of two important components. The first component name is called core enzyme. This core enzyme is responsible for the protein synthesis, RNA synthesis. Okay, the core enzyme is responsible for the synthesizing of RNA. The second component is called sigma subunit component, and this sigma subunit component is responsible for the recognition of promoters. Okay, recognize to identify the promoters. The sigma subunit component is very important. Hope you understood this point. Then one more point also there. Okay, in a transcription unit, the RNA polymerase enzyme open the DNA molecule. Okay, open the DNA molecule. Then it transcribe. Okay, open the DNA and form a transcription bubbles. Okay, and form a bubble-like structure. This bubble-like structure is called transcription bubbles. So, RNA polymerase enzyme open the DNA molecules and form a bubble-like structure. This bubble-like structure is called transcription bubbles. Understand of you? So, hope you are understood this point. Then, next point. Now, the core enzyme. Okay, as I told you, the first component of this RNA polymerase. This core enzyme move ahead. Okay, the core enzyme move ahead and manufacturing the RNAs and leaving the sigma subunit. Okay, the core enzyme of this RNA polymerase moving, manufacturing the RNA and leaving the sigma subunit. Understand of you? Understand? So hope you are understood this point. Then the next point is the end of a gene is marked by a terminator sequence. Okay, the end of a gene is marked by a terminator sequence and forming a hairpin-like structure. Okay, now the last point of this transcription unit is the subclass of terminators need an additional protein. Name of that protein is called RHO protein. Understand of you? Understand? Dear students, now we completed the first parts of this transcription process. Okay, the structure of a transcription unit. So here, maybe one more questions and two more questions also there. Okay, so hope you all understood these concepts. Then, now only we are going to study about the process of transcription. Listen very carefully, dear students. This is very, very important 5-mark question. Process of transcription. See, actually, In prokaryotic organism, there are three major mRNAs are there. Okay, three major mRNAs are there. mRNA, then tRNA, then rRNA. Okay, mRNA, tRNA, rRNA. So these are the three RNAs playing a very important role in the prokaryotic organisms. So all these three RNAs are very important to synthesize the protein molecule in the cell, in a cell of a prokaryotic organism. Understand? Here, the first enzyme, mRNA. Okay, the first RNA is mRNA. This mRNA transcribe one enzymes. Okay, this mRNA provides a template. That means it acts as a template. Okay, so there are three mRNAs are there: mRNA, tRNA, and rRNA. Here, this first RNA, mRNA, provide the template. It acts as a template. Okay. The next RNA, tRNA. This tRNA giving the amino acid molecule for synthesizing protein. You all know protein is made up of amino acid molecules. So here, this tRNA bring the amino acid molecule for synthesizing the protein. Then next one is rRNA. So this rRNA catalyze the transcription process, increase the speed of the transcription process. So all these RNAs playing a very important role in the production of protein in a cell, in the prokaryotic organisms. Okay, then next point about this process of transcription is, already I told you, one enzyme, DNA dependent RNA polymerase. So the single DNA dependent RNA polymerase enzyme can catalyze the polymerization reaction of all these three RNAs. Okay, so all these three RNAs are catalyzed. The transcription process or the polymerization process of all these three RNAs are catalyzed by a single enzyme. Name of that enzyme is called DNA dependent RNA polymerase enzyme. DNA dependent RNA polymerase enzyme. Okay, hope you are understood this point. Then the next point is, say this enzyme bind the promoter region of a DNA molecule. Okay, promoter region is the first region of the DNA. Okay, so first region in a transcription unit of a DNA molecule. So here this DNA dependent RNA enzyme, DNA dependent RNA enzyme, RNA polymerase enzyme binding the promoter regions only. Okay, then after the initiation of this transcription process, polymerase enzyme elongate the RNA molecule. Polymerase enzyme elongate the RNA molecule. So once again repeat, after the transcription process is started or initiated, the polymerase enzyme elongate the RNA molecule and adding the nucleotides one by one. Okay, so this enzyme add many number of nucleotides one by one. Thus, it increases the length of the RNA molecules. Thus, it increases the length of the RNA molecule. Okay, understood? Then, next point is, when the RNA polymerase reach in the terminator region, all the RNAs, the nascent RNA and RNA polymerase synthesis, everything are stopped. Okay, so when this RNA polymerase enzyme reach the last region, that means the terminator region, automatically the nascent RNA means here the RNA which is elongated, that RNA is falls off. That RNA is fall off. Hope you are understood this point also. Then the next point about this transcription unit is, say, maximum in this transcription process, the RNA polymerase enzyme, so this one enzyme only playing a very important. Okay, so the RNA polymerase enzyme involved in the elongation process, initiation, elongation and termination. These are the three steps involved in the transcription process. Okay, initiation, elongation and termination. Here, this RNA polymerase enzyme controlling the elongation process. Okay, controlling the elongation process. And the initiation process and the termination process both are controlled by two more factors. Okay, listen very carefully. See, the initiation factor is controlled by a factor called initiation sigma factor. Initiation sigma factor. So this factor only controlling the initiation step. Then the termination. Okay, stop the last step. Termination step is controlled by another known factor. Name of the factor is called termination factor RHO. Termination factor RHO. So initiation, elongation and termination. These are the three steps. Here the elongation process is controlled by RNA polymerase enzyme. Initiation is controlled by initiation sigma factor and the termination is controlled by termination factor. Okay, termination RHO factor. Understand of you? Here the RNA molecule associate these two factors and instruct the RNA polymerase enzyme whether the process is initiated or terminated. Understood? I hope you understood this point. So the RNA molecules are associated or connected with these two factors, this initiation factor and termination factor, and instruct the enzyme. Which enzyme? RNA polymerase enzyme. Okay, so finally the RNA polymerase enzymes only taking a decision whether this process, transcription process, is initiated or it should be you have to terminate. Understand of you? Hope you are understood this point. Then in bacterial cells, sometimes the termination process and the elongation process takes place simultaneously. In bacterial species, the termination, transcription, not termination, sorry, the transcription and translation. Translation meaning is the next step. Okay, transcription meaning already I told you, the copying of genetic information from one strand of the DNA molecule to RNA is called transcription. Translation means the process of production of proteins from the RNA is called translation. Okay. But in bacterial cells, so first the transcription process takes place, then next only the translation process takes place. It's happening now in a normal cell. But in bacterial cells, sometimes the transcription and translation process takes place simultaneously. And sometimes the translation process begins first. After that only the transcription process takes place. The reason for this condition is, in bacterial cell, the genetic materials are not separated from the other cell organelles by the nuclear membrane. Okay, in bacterial cell, there is no particular nucleus. So, in bacterial cell, the genetic materials are not separated from the other cell organelles by a nuclear membrane. So, because of this reason, the translation process is first takes place than the transcription process in some bacterial cells. So, hope you are understood this point. Okay, so now we completed this point. Okay, then next point. In eukaryotic organisms, there are 3 RNA polymerase are there. In eukaryotic organism, there are 3 RNA polymerase enzymes are there. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Okay, here this RNA polymerase 1 transcribe rRNA. RNA polymerase 2 transcribe hnRNA, involved in the transcription of hnRNA. Then RNA polymerase 3 involved in the transcription of 5sRNA. So hope you understood this point. In eukaryotic organism, there are 3 types of RNA polymerase are there. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Here RNA polymerase 1 involved in the transcription of rRNA. RNA polymerase 2 enzyme involved in the transcription of hnRNA. And RNA polymerase enzyme 3 involved in the transcription of 5sRNA. So, hope you are understood this point also. Understand of you? Then, in eukaryotic organism, the monocystonic structure. Already I told you there are 2 types of structural genes are there. One is monocystonic and next one is polycystonic. So, in eukaryotic organism, the monocystonic structural gene have some interrupted coding sequence, some barrier coding sequence. So these sequences are called exons. Once I repeat this point, in eukaryotic organism, the monocystonic structural gene have some interrupted coding sequence. These coding sequences are called exons. And the non-coding sequence also there. Here these non-coding sequences are called introns. So exon means interrupted coding sequence and the intron means non-coding sequence in a monocystonic structural gene. Understand you? Here The introns are removed by a process. Name of this process is called splicing. Name of this process is called splicing. Okay. Then in HNRNA, two more additional process also takes place. In HNRNA, two more additional processes takes place. The first process is called capping and the second process is called tailing. Here capping means adding of one important substance, methyl guanosine triphosphate at the 5' end. So capping meaning the adding of methyl guanosine triphosphate in the 5' end. This process is called capping. And tailing means adding of adenylate residues at the 3' end. Adding of adenylate residues at the 3' end. So once again I repeat this point. In HNRNA, two more additional processes also takes place during the transcription. One is called capping and second one is called tailing. Here capping meaning adding of methyl guanosine triphosphate at 5' end and tailing meaning the adding of adenylate at 3' end. Okay. So after the completion of these two processes, capping and tailing, the HNRNA is now called mRNA. The HNRNA is now called mRNA. So hope you all understood this point. Then next point about this transcription is, see, actually the exons may have many polypeptide chains. Okay, the exons may code for many polypeptide chains. Okay, and since the arrangement of exons and the removal of introns are flexible, okay, listen very carefully. Since the arrangement of exons and the removal of introns are flexible, many exons are joining together, or many exons are combining in different ways. Okay, combining in different ways to form new genes. Once again, repeat this point. Since the arrangement of exons and the removal of introns, these two processes are very flexible. Okay, very flexible. But the many introns are joining togetherly. Okay, sorry, many exons are combining togetherly and forming a new genes. Understand a few? Then next point. A single gene can produce different proteins. Okay, listen very carefully. A single gene can produce different proteins by arranging their exons, okay, in different ways. Okay, actually it's a single gene producing single proteins, but sometimes a single gene can produce different protein. How it is producing different proteins? The single genes arranging their exons. Okay, arranging their exons in different ways. Okay, that gene arranging their exons in different ways, and finally that single gene can produce different proteins. Okay, so hope you are understood this point. Then next point, see, actually the introns can form before or after it can form before or after the evolution of eukaryotic gene. Okay, so the introns, it can form, okay, but it can form inside a eukaryotic gene, but it can form before or after the evolution of eukaryotic genes. Okay, here the introns are mobile genetic elements. Okay, mobile genetic elements means it can act as a mobile transposons. And it can join themselves inside or outside a target place. Listen very carefully. Introns are mobile genetic elements. That means they can join themselves inside a place, inside a site or outside a site, outside a particular site. Okay, and it can act as a mobile transposons. Here mobile transposons meaning transfer of genes between some organisms. Okay, transfer of gene between some organism, and this is called HGT, horizontal gene transfer. This process is called horizontal gene transfer. That means these introns can transfer between two organisms. Okay, between two eukaryotic organism, or between two prokaryotic organism, or between one eukaryotic organism and one prokaryotic organisms. Understand a few? And this horizontal gene transfer playing a very important role in the evolution of life. Understand, my dear students? So I repeat the last point. Introns are mobile genetic elements. That means they can join themselves inside or outside a target site, and it can act as a transposons, mobile transposons. That means it can transfer genes between the organism, and it is called HGT, horizontal gene transfer. That means the gene can be transferred between two eukaryotic or between two prokaryotic organisms, or between one prokaryotic and one eukaryotic organism, and this intron playing a very important role in the evolution of life. So dear students, now we have completed one very important topic, the process of transcription. The process of transcription, and here also many 2 mark questions and many 1 mark questions also there. So, okay, so you all must study this question answer. It is very important. Okay, so we can see in the next class. Okay, thank you.